Welcome to Planetterrian Daily, episode 96, coming to you on June 20th, 2026. Here's what's new in science, health, and longevity research. Prion proteins may hide short peptides that kill drug-resistant bacteria, AI analysis shows. University of Pennsylvania scientists used AI to scan prion and prion-like sequences and identified short prionin peptides with antibacterial activity against resistant strains. The peptides appear embedded within proteins best known for causing neurodegeneration. Structural modeling suggests the antimicrobial segments are normally masked until proteolytic release. Follow-up work will test efficacy and safety in animal infection models. The Planetterrian Spotlight looks at how this AI scanning of prion sequences uncovered short, embedded peptides with direct antibacterial activity against resistant strains. The finding reframes prions as potential reservoirs of innate defense molecules rather than solely pathogenic entities. It opens a route to mine neurodegeneration-linked proteins for new antibiotic scaffolds. Next, researchers will synthesize and test the most promising prionins in live infection models. Which other protein families might hide similar overlooked functions? Now, shifting to a very different area of research. A review in Nature Reviews Endocrinology outlines how Epstein-Barr virus, already linked to higher multiple sclerosis risk, appears to confer protection against autoimmune type 1 diabetes. The opposite directional effect on two autoimmune conditions points to virus-specific modulation of immune tolerance. Researchers note the established multiple sclerosis association provides a useful comparator for dissecting mechanisms. Further human cohort studies are needed to confirm the protective association and identify involved pathways. Now, here's something most people get wrong. You've probably heard that hormones such as cytokinins regulate growth only in plants. In reality, these same molecules are now detected in mammalian cells and appear to influence stress signaling and metabolism. Right now, as your cells manage daily oxidative and nutrient stress, trace cytokinins from diet or resident microbes may be modulating the same pathways plants use to slow aging under drought. A single cytokinin molecule can alter gene expression across dozens of targets, illustrating how a small concentration produces outsized effects. The next time you drink green tea or eat certain vegetables, consider that you are also ingesting compounds once thought irrelevant to human biology. Watch for upcoming studies that measure cytokinin levels in human blood and link them to measurable changes in inflammatory markers. On a different note, Caltech researchers showed that stressed soil bacteria remodeled gene expression and metabolism to cope with phosphorus scarcity, altering interactions with plant roots. The changes affect nutrient cycling and plant growth under nutrient-limited conditions. Experiments combined controlled microcosms with transcriptomic profiling of bacterial communities. The findings matter for predicting soil responses to changing climate and fertilizer use. Next up, a Freie Universität Berlin analysis emphasizes that fungal networks move carbon through soil food webs and stabilize plant communities under stress. Mycorrhizal and saprotrophic guilds together influence both agricultural yields and long-term carbon sequestration. The review synthesizes field and laboratory evidence across ecosystems. Future work will quantify how management practices alter fungal contributions to soil carbon, from below-ground fungal networks to root-associated microbes that specifically aid heat tolerance in crops. Rhizosphere microbiome surveys suggest certain bacterial and fungal taxa correlate with improved crop performance under elevated temperatures. Inoculation trials are testing whether selected consortia can be transplanted to boost resilience. The work focuses on microbes that influence water uptake and hormone signaling in roots. Field validation across multiple growing seasons is underway. from beneficial microbes in agriculture to an animal virus that revealed unexpected tissue preferences during spillover. Early U.S. cattle infections with H5N1 produced severe udder disease while largely sparing the respiratory tract, delaying recognition that the virus had jumped species. Diagnostic sampling focused on milk rather than nasal swabs proved more sensitive. The altered tissue tropism differs from patterns seen in other mammals. Surveillance programs have since adjusted sampling protocols accordingly. From emerging pathogen behavior to engineered nanobubbles that address environmental contaminants. Nanobubbles, roughly 100 nanometers across, generate reactive species that break down oil, algae, and other contaminants without leaving chemical residues. Wen Zhang's team at New Jersey Institute of Technology is testing the approach in field-scale water treatment systems. The bubbles remain suspended for long periods and deliver oxidative power directly at pollutant interfaces. Next steps include scaling generation methods for municipal and agricultural use. University of Copenhagen economists tracked Danish firms facing low-cost Chinese imports. They found that while offshoring reduced local emissions, overall global emissions increased. The net rise occurred because production shifted to regions with higher carbon intensity per unit output. The study used firm-level data matched to trade and emissions records. Policymakers are examining whether carbon border adjustments could alter this pattern. An international team from Tohoku University, Tokyo University of Science, Vanderbilt, and Adelaide developed an ambient air method. They used it to synthesize stable 15-atom iridium nanoclusters. The clusters delivered 1.5 times the mass activity of standard iridium catalysts. The clusters ran without degradation beyond 20 hours. Precise size control was achieved without high temperature or vacuum equipment. Researchers aim to extend the approach to other precious metal catalysts. JWST observations led by IPCAC at Caltech revealed a massive galaxy cluster. The cluster acts as a strong gravitational lens at a redshift where such structures were not expected to be fully assembled. 3 papers detail the cluster's concentration and lensing strength. The data challenge current models of structure formation during cosmic noon. Additional JWST pointings are planned to map member galaxies and dark matter distribution. Researchers generated a high-resolution atlas of the inflorescence meristem in Arabisox, identifying 18 transcriptionally distinct cell populations that give rise to stems and flowers. Single-cell RNA sequencing traced lineage trajectories from stem cell niches outward. The map clarifies how positional information is translated into organ identity. The dataset will serve as a reference for crop species with similar meristem organization. University of Technology Sydney physicists demonstrated that rotating atomically thin hexagonal boron nitride flakes lets them tune the emission properties of embedded quantum emitters. The twist angle alters local strain and dielectric environment, shifting photon energy and coherence. Experiments combine optical spectroscopy with precise mechanical rotation stages. The method offers a route to integratable quantum light sources for photonic circuits. Psychological experiments link heavy platform use to weakened sense of body ownership, with participants reporting reduced alignment between visual appearance and felt bodily self. The effect appears tied to repeated exposure to edited images rather than simple screen time. Researchers measured proprioceptive drift and explicit ownership ratings after controlled viewing sessions. Longitudinal studies are planned to assess reversibility. A survey of 463 German adults found that familiarity with plants and birds, typical of agricultural landscapes, drops sharply in younger cohorts compared with older respondents. Common species such as dandelions remain widely recognized, while meadow specialists do not. The pattern points to reduced direct contact with farmland biodiversity. Educational programs targeting regional species identification are being developed. New findings continue to show how basic biology and environmental interactions shape health in measurable ways. Keep an eye on the animal infection model results for the synthesized Prion peptides. That's Planetterrian Daily for today. If you enjoyed this, a rating or review on Apple Podcasts or Spotify really helps new listeners find the show. I'm Patrick in Vancouver. Thanks for listening, and I'll see you tomorrow. And before you go, this show is part of the NARA Network, a family of daily podcasts covering tech, science, markets, and more. If you enjoyed today's episode, give SpaceX Daily a listen, the daily companion for following SpaceX as a public company from Starship to the stock ticker. You can explore the whole lineup at naranetwork.com. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.